All right, so yeah, thanks for the introduction, Nick. So greetings and salutations. I'm Nabil-Fareed. I'm a bioinformatician at the Quadram Institute, and we are one of the sites that contribute to COG UK. I started with SARS-CoV-2 about a year ago now, and that's involved setting up our bioinformatics pipelines, collating metadata, going to analysis, and then submitting our data to our central repository within the consortium. And then also doing a bit of data submission for public databases of ENA and GISAID on my own. And so I represent an average bioinformatician with some practical experience, and today I'm going to share that with you. And so you can learn from some of the hassles and mistakes that I've encountered or we've encountered along the way. So hopefully by now you have a basic understanding of the workflow and of sample preparation, sequencing, and analysis as presented by Josh, Will, and Matt. And I won't repeat the workflow in detail here, but I will include some online resources and publications which you can review later. What I will talk about today covers important caveats I've encountered or heard from others, and I've put a summary of all of them here. I'll quickly introduce them and give you some tips on how to actually address them. I'll first talk about these problems in terms of sequencing data and then in terms of contextual metadata. The specifics around phylogenetic analysis is actually all in the next session, so I won't say anything about that here. So in terms of sequencing, the most important thing to remember is SARS-CoV-2 is a bit different to any other biological agent you've worked with. You can't directly use the same approaches that you've been using in, say, human or bacterial genetics. If you're following the tiling amplicon approach implemented in Arctic, that also introduces its own peculiarities that you need to take into account. Well, some of which was touched on by Josh earlier. The second thing to remember is SARS-CoV-2 isn't particularly special either. It doesn't have a very complex genome. And I think after 1 year, we've sort of exhausted all the different analysis approaches that you could attack it with. For instance, I myself tried to dig deeper into the basis of the variant calling for SARS-CoV-2, and I tried to calculate different metrics of all the mapped read data. And I thought I was very clever. This is actually a similar question to what someone asked Matt just before. But all what I was doing was totally redundant because actually all the information I wanted was produced as part within the Arctic bioinformatics workflow and also in another tool called NCOV tools as well. So I've just put this table up here showing you all the different pipelines that you have available. There are many, many more available. You might find that a lot of these are actually based off the original Arctic pipelines. And if you want to see a lot more, you can go to the link down here on the bottom. There's different pipelines for not just the tiled amplicons, but for metagenomics, for ion torrent data, for Sanger, capillary sequencing, whatever you like. So do have a look at that, and there is probably something already existing for what you're trying to do. That being said, I think one area that tends to require bespoke solutions is some of the more in-depth technical analysis where you require a specific interpretation. So if you're talking about the spread of COVID across a country, you're going to describe that and illustrate that a bit different to, say, a local ward outbreak. And then this again will depend on the audience and the exact situation. The questions that we're fielding now, for instance, in terms of dealing with the new variants is a little bit different to what we were doing even just over Christmas. But these questions are usually changing week to week, at least in my experience. So this again isn't like an avenue for tool development, but more that you need to be flexible with standard data analysis tools like R or Matplotlib or ggplot or D3JS or whatever and so on. So basically, for any given task, there probably is a specific SARS-CoV-2 pipeline that already exists. Obviously, what's available through Arctic is the best, and you should really try to use those first. You may find that there's some specific one-off questions that you'll need to answer, but again, you're going to be using standard data analysis and statistics tools. Now, I say all of this because there have been some really horrible examples where people have just used tools that they're used to and they didn't understand their data or the literature. One of the simpler issues I've seen is some folks tried to do a straight de novo genome assembly of amplicon data, which doesn't make any sense. For one, the read still included the synthetic primers. So the assembly turned out into this weird mismatch of viral reference genome based off the primers and then the sample that they're looking at. So in this case, we've heard it from the other speakers, but it's important to remember that you need that extra step of filtering and masking those primer sequences before you really start to use it in your variant calling. But again, this is handled by the SARS-CoV-2 specific pipeline. So it's more a question of understanding what these tools are doing and then just sort of leaving it at that. Another more complicated example is people try to adapt a practice that's actually quite common in human and bacterial genetics, where you map reads and then call variants in areas, call variants. And then in areas where you have gaps, you backfill with the reference genome. Other times people might do something like they take the reference genome and then they call variants using some data, and then they take those variants and just sort of mutate the reference with those changes. Some other clever people might try to use ancestral reconstruction to impute missing bases. And in all the three of these cases, if you do this, do not upload this to public databases as if this was a complete run-of-the-mill SARS-CoV-2 genome. This is simply not what people are expecting. This is not common practice. Do not do anything like this. Moving on to a more simple matter, but equally important one, is you need to make sure that you have removed human reads from your data. Obviously, your SARS-CoV-2 samples are coming from humans mostly, and some of the host genome might be sequenced. So it's imperative to actively remove these reads before depositing the data in public archives. Protect the host, particularly since many of the samples probably are unconsented. Now, even though with ARTIC, you're still going to be using targeted sequencing, but we've found that some hosts might still sneak through. So it's best to double check on the bioinformatics side. The simplest solution is just a simple workflow like this, where you just map all the reads onto the SARS-CoV-2 reference genome and then remove all of the unmapped reads before public release. There are other methods like the Krakens and so on and on if you want to try that. Another thing to consider is the initial viral load in the sample, which will impact the utility of the data. So if you have too little virus, so that's high CT in a sample, there will not be enough material to generate a phylogenetically useful genome, no matter how much you amplify it. And knowing this in advance can help you pre-filter samples if they are unlikely to assist your research question. So the current exact cutoff tends to vary a little. So this isn't a hard and fast rule, but once you get started with your own samples, you will figure out what your cutoff is pretty quickly. In our study of our local area, we found a CT over 32 is probably a no-go, which is illustrated here with this plot where you've got the CT value on the x-axis and then the percentage of uncalled bases on the y. And you can see a rapid increase in the number of missing bases over a CT of 32, at least for us. You should remember that for your phylogenetic analyses, you're aiming for 90% of the bases to be called confidently. And when I say confident, that means you need over 10 times coverage for each base on Illumina and over 20 times coverage for each base on Nanopore. You may have other research questions where these samples with lower yields might be useful, and that's entirely up to you. What do you do with those? But these are the cutoffs if you're going to start using phylogenetics that you need to be aiming for. Okay, so always using control samples. This is pretty obvious, but it seems to be overlooked sometimes. Controls matter, and every sequencing experiment should contain negative controls at a minimum. But not only that, they should also be bioinformatically analyzed to quantify the level of contamination introduced to the preparation and sequencing process. A human person should also manually check the contamination profile at this point before proceeding. Now, I have to stress this because SARS-CoV-2 sequencing has a number of peculiarities that makes this really important. on the one hand, you're heavily amplifying your starting material. And I mean heavily that if you're doing Illumina sequencing, we regularly see 1,000 times coverage up to 3,000 times coverage. And Josh mentioned the issue of barcode hopping and the variation of primer efficiency. And that together can cause a lot of confusion. To make matters worse, the SARS-CoV-2 genome itself has very low diversity. And so even if there's a small amount of crosstalk between lineages, that may radically alter your results. So this is just some plots of that, this is a fairly common example for us. It's pretty clear cut of which one's the negative control and which one's the sample. This is just the x-axis is just the length of the SARS-CoV-2 genome, and then the depth of reads mapping to it on the y. But even in this case, which is pretty obvious what's going on, you can see that some reads map to the blank. And you can see in the actual sample, there are some regions that couldn't be called, which are marked in red. Okay, so I've had a few, and even now we just had some questions around quality control. And I thought I would put this table up with some of the problems that you might run into and some of the solutions you can use to address those. I think, again, that the final mapped BAM file that you get out of the analysis pipeline has a lot of information. And just being good with SAM tools, you can solve a lot of different problems that way. For instance, if you're worried about primer dimers and you want, or maybe you want to see if certain regions are dropping out for one reason or another, using a combination of SAMTools depth, SAMTools pileup, and understanding the cigar strings will go a long way into helping you figure out what's going on. Another thing you can do is keep an eye out for cross-contamination, which I mentioned before. And one general check for errors is you can check if there's any weird magical frame shifts that have suddenly shown up in a sample. GISAID has a service where you can compare against, take your sequence and compare that against all available genomes in the database, which is really helpful. So you might have a particular genome that you're a little unsure about, and you might suddenly find that it has all these new and novel, never seen before frame shifts. And that's a pretty bad sign that something has gone wrong. Some potential issues that I've described, there are plenty more issues that you can run into, and some of the issues might be more pertinent than others. It's really dependent on your setup, on what you want to go after. I don't think there is an all-spanning QC regimen that you could employ. All right, so that's it in terms of the sequencing side of things. So let's switch over and talk about some of the contextual metadata that encloses your sequencing. Now, having good metadata is really important. A sample with no context about where it was from or when or from who is totally useless. You need to know these things to be able to say anything informative. But the frameworks to collect and share this data are a little bit more nebulous than dealing with genomes. So to help out, we put together a manuscript, the links up here, which will take you through some of the considerations when you start collecting metadata. One of the things I will mention is that it's important to think about what kind of information you want to collect and which information would be most useful. And in developing this particular manuscript, we went through a lot of consultation to figure out which fields people were collecting and why. And so that'll give you a good sort of primer of what you could possibly be looking at. If you're also simply wondering what information is required for you to submit your data to your genome to a public database and in what format they're expecting, have a look at this manuscript and it'll take you through all of that as well. What I will add is that the information that you collect doesn't have to all be made public just because you have it. In fact, the information that needs to be shared publicly is quite limited. You can get away with not giving a lot of information. And what I found that if you understand this and explain this to collaborators, this helps them be a lot more forthcoming with information. Often when they're frightened about giving you information about clinical samples, it's often due to a lack of clarification. And that's what this figure shows here. It just shows that there's a total pool of data or information that we know about a sample, and it often needs to be very clear about which information will be shared amongst the group only, which will be made public, which will be shared with collaborators, and so on. And if you say that up front, people will be will tell you a lot more about your samples, and then you'll be able to get a lot more out of it. So do have a look at this particular manuscript if you're getting into collecting contextual sample data. Now, following on from that, there's an obvious question of how do I actually implement good data management for my SARS-CoV-2 sequencing project? And there's a lot of ways to do it. Now, initially, you might be just sending emails with spreadsheets to and from the clinic. And over time, you may graduate to more complex systems like a full website with user accounts and a relational database. It really depends on your situation, what level of infrastructure you require. I will say that the more complicated your setup, the more upkeep and overhead you're going to have to invest into looking after it. And what I will say is that you can manage pretty well with just sharing Excel files, but you have to remember one little secret. You need to have good validation of the data as it's being created and modified. You need to have simple systems that check that the new samples being added have sensible information. And that applies to whatever level of data of infrastructure you use. The other thing that you need to consider is that you're going to be dealing with Microsoft Excel or similar spreadsheeting program. Even if you can somehow avoid it within your own ecosystem, people are going to send you data, or they're going to expect you to report data in that file's format. So your system needs to cater to that. In the end, what I think is more important is you start thinking about what would a spreadsheet from the clinic say? What would it actually look like? What information would it contain? And then how would you check that that information is correct? You should then implement these checks as like a simple programming script that can read either delimited text or Excel sheets. There's a lot of libraries available that will help you do this efficiently. I mainly use Python, and these are the ones I use here listed on the slide. Having this validation available would allow you to run it on new data as you receive it, for instance, or you could periodically check your existing data with it. But it's only through some kind of process like this can you ensure data integrity. Now, if you're interested in building a central database infrastructure, I recommend that you have a look at the Majora preprint, which is the link here. And this is the system that's used within COG UK. And a lot of these issues are described in a lot more detail, and there's other aspects that are introduced there. I think as part of the homework, I've created some sample data, and you'll need to come up with a strategy for identifying errors in metadata. But for now, let's just move on. Okay, so using a good sample identifier. This may seem simple, but this is actually quite difficult to do well. The sample ID that you choose is actually very important. This is the name that's going to show up on pipeline output files. It's going to be the file names for BAM files. It's going to be in reports. It's going to be on figures. You're going to have it in a table, and you're going to refer to it amongst colleagues on video calls. So using this particular ID. So you need that ID to be easy to read by a human and a computer, and you need that ID to be unique. And I will tell you from experience that it is immensely difficult to change the ID from one format to another halfway through a project. So I recommend that you come up with a good naming scheme and use that throughout. Now, you may be inheriting IDs that are generated from elsewhere, say the clinic that did the sampling have given these things names, and you need to then consider if these IDs are something that you're willing to use throughout your project. Now, I would advocate avoid making lots and lots of different identifiers. But if you're really pressed, if the IDs they're giving you aren't really up to scratch, then you should introduce a tandem internal identifier as well to sit alongside that one that's a lot more sensible. It's also a good idea to think about printed sample labels as well, which you can use throughout the lab work. This requires a certain degree of infrastructure. COG UK, for instance, uses these pre-printed barcodes, which again is described further in the Majora preprint. And I'll give the full link for further reading material. Okay, so this is the last snippet from me. Data submission is very important. By sequencing SARS-CoV-2, you're contributing to a global effort, and your data will be used by others for the purpose of getting this virus under control as quickly as possible. You need to provide your data in a way that someone else can understand it. Now, you should be depositing your raw data and genomic sequences in public archives. So in that regard, you should consider submitting both to GISAID and the INSDC. So that's your EBI and CBI or DDBJ. This is a rapidly changing field, and by making the data open, by allowing reanalysis, this allows new questions to be answered that hadn't been thought of originally. Now, of the two databases, GISAID is probably the easier of the two to use, but it only accepts the final consensus sequence, and really you should be uploading the sequence reads as well, and you can submit both to any of the INSDC databases. In both databases, it takes some time to register and have your accounts activated. And it takes even longer to try and get used to the submission system. So if you are sequencing SARS-CoV-2, I recommend that you set up your accounts as soon as you can. Do not leave it to the last minute. The details of the submission processes themselves are actually pretty involved, and I won't go into it here. But again, some of the information is available via this preprint. One thing I will mention, that if you are depositing to both GISAID and the INSDC, you should cross-reference the accession numbers. So your INSDC record, your GenBank record, should have the GISAID ID included as part of the metadata. We include a step-by-step guide of how to do the data submission in the contextual data specification, which is available in the preprint in this highlighted link. Yeah, so here are all the readings that I mentioned throughout the talks, which you can go back to later. I'll post these slides and all these links in the Slack, so you can have a look at those. And yeah, that's it from me. I should thank Andrew and the rest of our team at QIB. A lot of the issues that I'm talking about are things we all worked through together. Some of the few initial gotchas that I talked about came through some discussion with Duncan, so many thanks to him. I should thank the COG UK people. A lot of them are actually here as part of the workshop, and it's really become its own community, and I've learned a lot as we've dived into COVID genomics together. Thanks to Quadrum, who actually allow me to work on COVID. This isn't actually my day job. I'm supposed to be working on foodborne microbes, but here we are. And thanks to CLIMB-BIG-DATA and ARTICnetwork for having me along today.
Brilliant. Thank you so much, Nabil. Give him a round of hearts or whatever you want. That was really a great way to end the session. And I think some really useful advice there in terms of quality control of data, and also the important thing about sharing, data sharing, absolutely vital for this to work. Nabil, how do we check the quality of data in public databases?
Okay, so there is some quality check that's built into the GISAID database. If you ever go to that, they do a quick check, sort of kind of things along the lines of looking at frame shifts or the number of called bases. So that's a good place to have. So part of it's done for you. Obviously, the best way is to pull down people's BAM files and consensus sequences and apply the same QC stuff that I was discussing during the talk as well.
Yeah, and I think that emphasizes your point about submitting read data as well as consensus data, doesn't it? Because, as you say, GISAID only supports consensus. And I mean, maybe you could comment on just the difficulties, because there are a huge number of groups globally doing coronavirus sequencing with a large number of different laboratory protocols, although I think the ARTIC protocol is by far and away the most popular one. There are many different types of protocols being used, as Josh mentioned, including metagenomics and Bates. And then actually quite as you mentioned, quite a wide diversity of different pipelines. And, you know, these might apply different types of cutoffs, depth cutoffs, quality cutoffs on a local center by center basis. So, you know, how can that be reconciled really? And, you know, how can we guarantee quality across all this diversity of approaches?
Yeah, I think if you, a lot of it will shake out during the phylogenetics. I mean, obvious things like if one sample is suddenly on the other side of the planet in terms of branch lengths, it sort of sticks out like a sore thumb. So aligning it together and looking for consensus with all of them helps. There's very little you can do beyond that if all you have is a consensus sequence, because you don't know, you can't, even if it's described what people have done, you can't go back and verify it without the raw reads. So that's, yeah, as Nick's saying, definitely what's available for raw sequence data is really, really helpful for going back through that. I lost my train of thought because Matt said something.
I think that's right. And I think in the COG project, we made sure that raw reads are available for all of the genomes. One thing that
I've remembered. If what I found is that the description for the pipelines, if your ARTIC is very standardized, so that's easy. That's very easy. If someone says they've used that, you kind of know what they've done. But if people do something fuzzy and weird, you are probably going to have to go back to their raw data and then redo everything in a standardized fashion. So you'd reanalyze your entire data set of their data and your data together using one approach to make sure that it's consistent.
And I think we would like to, as part of the next phase of the CLIMB Big Data project, make it much more easy for people to access underlying raw read data, you know, things like variant frequencies directly from the BAM files, so that in the situation where you really are concerned about a particular mutation, and whether that, if your hypothesis hinges on the presence or absence of mutation, you can look at the read level evidence for that base call. I think that in some cases that's important, but in the more, as you say, in the more kind of global approach, looking at all the data together, a phylogenetic approach is often the best way of tossing out low quality sequences because of strange positioning in the tree or long branch lengths or other types of phenomena like that.